Bismillahirrahmanirrahim. Let's start our today's class for the preparation of scientific officer of plant breeding and genetics. Our today's class number is 13. Questions are MCQ types and this class is managed by EPSR. Let's move towards our first question, and first question is that different classification of different isolating mechanisms had been given by Dobzhansky. Dobzhansky gave the different classification of different isolating mechanisms. A point mutation in which a purine is replaced by a different purine or pyrimidine by a different pyrimidine is called transition. A point mutation in which a purine is replaced by a different purine or pyrimidine by a different pyrimidine is called transition. A genetic unit capable of moving from one chromosome site to another or from one replicon to another is called transposons. Transposons. Next question is that what is the polyploidy level of triticum aestivum? And the triticum aestivum ploidy level is 6x. It means triticum aestivum is a hexaploid. A point mutation in which a purine is replaced by a pyrimidine or vice versa is called transvariant. Transvariant. Next question. If standard deviation is 9, then the variance will be 81. And next question is that dash is the study of heredity and variation. Genetics is the study of heredity and variation. Germ theory was given by August Weismann. A sudden heritable change is called as mutation. Mutation means sudden heritable change or genetic change and mutation. Next question is that fitness of a genotype to a given environment is called its adaptability. Adaptation of a genotype to its new environment is called adaptability. An RNA virus that uses reverse transcriptase to assemble a DNA copy of its RNA genome is called retrovirus. Retrovirus. Next question. The DNA form of an RNA virus that can integrate in the chromosome of the cell is called provirus. Next question. An assembly of ribosome active in translation and connected by the some messenger RNA strand is called polysome. Polysome assembly of ribosomes active in translation and connected by some messenger RNA strand. Next question. The suitability of a genotype for journal cultivation over a wide range of the environment is known as stability. Combining ability analysis provide information about gene action and good journal combiner and good specific combiner. All these three factors are necessary or used during the combining ability analysis. Additive gene action refers as additive variance and additive additive multiplied by additive epistasis. This is the additive gene action. Next question, that the segment of DNA associated with one or more structural gene which interact with the product of a regulatory gene in the control of transcription is called operator. This is known as operator. Next question, that dash is called the father of modern genetics, and modern genetics father is called George Mendel. First segregation generation is F2. First segregation generation is F2. Next question. Phenotypic ratio in F2 generation for monohybrid cross is 3:1 is the correct answer. Next question. Genotypic ratio in F2 generation for dihybrid cross is 1:1:2:4:2:1:1. This is a genotypic ratio for F2 mm, dihybrid cross, which is shown in F2. Next question, a virus that causes lysis following its multiplication in the host cell is called lytic virus. Uh, Other question, the genes which code for essential function basic to all cell types are known as housekeeping genes. A gene which code for essential functions basic to all cell types are called housekeeping genes. And the next question, the percentage of variation seen in expression of a character that can be attributed to genetic factor is called heritability. Percentage of variation seen in the expression of a character that can be attributed to genetic factor is called heritability. Which of the following book was authorized by D.S. Falconer during 1960? Is the book name is Introduction to Quantitative Genetics. Next question, which of the following design is called nested design? And the nested design is NCD1. NCD1 is known as nested design. Next question, which of the following design is the most powerful one? And most powerful one design is the NCD2. NCD2 is the most powerful design. The total of all the genes carried by a population at a given time is called as gene pool. Genes are present on chromosomes. As you all know that genes are always present on the chromosomes. Our next question is that an individual having single set of chromosome is called as haploid individual. And the next, as gene can exist in several alternative forms, and these alternative forms of genes are called as alleles. Next question, a gene affects more than one character simultaneously is called as pleiotropy. When a gene affects more than one character simultaneously, and this condition is called pleiotropy.